Thanks, Janet. So it's a great pleasure for me to be here this morning introducing the speakers, my friends and colleagues in the CRISPR field. And as a basic research scientist, I love the story of CRISPR. I think it's a beautiful example of the power of basic research and how curiosity-driven research can really lead to advances and new technological discoveries that we could never, ever have imagined. So CRISPR research initially was focused on discovering how bacteria protect themselves from infection by viruses. So bacteria, like humans, can be attacked by viruses that infect and kill them. And some of the early work in the field, really one of the seminal discoveries was in 2007, and that was work done by Rodolphe Barangau and Philippe Horvath. We heard from them yesterday, as well as Sylvain Moineau, who's speaking this morning. And what they showed was that bacteria have an immune system that helps to protect them against killing by these viruses. And it was only about half a dozen years ago, actually, that Sylvain then showed that this immunity that these bacteria have actually is provided by a protein that causes double-stranded breaks in the DNA. And so when the virus infects the bacterial cell, the bacteria has a way in which it can chew up the DNA and kill the virus. And in the few years after that discovery from Sylvain's lab, there were a lot of rapid advances in the field of CRISPR, and we heard from Jennifer Doudna yesterday, Emmanuel Charpentier, and Feng Zhang. And they made a lot of discoveries, and they took this technology and ran with it, and have been using it to develop tools for gene editing in human cells, bacterial cells, and all different kinds of cells. And I think, in the last couple of years, the technology has really exploded, and there's hundreds of labs around the world who are using this technology to edit human genomes, to develop new antimicrobials, and really from the initial beginnings, we never ever could have predicted where this technology is going to go. And so this morning, we have 4 talks from people who are going to be telling us about the technology and the work that's ongoing in their lab for it. First up is Sylvain Moineau, and he's going to give a historical perspective on CRISPR and, from humble beginnings to today's headlines. So Dr. Moineau is a Canada Research Chair in Bacteriophages, and he's a professor of microbiology at the Universite Laval. He got both his undergraduate degree in microbiology and his PhD in food sciences from the Universite Laval. He then completed an industrial postdoc in Florida with the food ingredients company Quest International, and at the time this was a division of the multinational Unilever Corporation. And during his time with Quest International, his research focused on the mechanisms that lactic acid bacteria use to defend themselves against viral infection. In 1996, he moved back to Canada and took up a position at the Université Laval as a professor. And he is a faculty member in the Department of Microbiology, and he's continued his research there in phage biology and resistance mechanisms in lactic acid bacteria. Dr. Moineau won the NSERC Synergy Award for Innovation in recognition of the work done in long-standing collaboration with AgriPure Dairy Corporation. He's been on Thomson Reuters' list of highly cited researchers, and he was recently awarded the Flavel Medal by the Royal Society of Canada for an outstanding contribution to biological science in the past decade. And importantly, Dr. Moineau is also the curator of the Felix Durrell Reference Center for Bacterial Viruses, and this is the world's largest collection of reference phages. And so we're going to hear from Sylvain right now.
Merci beaucoup, Karen. Bon matin à tous. Good morning, everybody. Thank you for being here. First of all, I want to thank the Gardner Foundation for their kind invitation. Fantastic evening last night. It was great to see everybody. I want to congratulate the winners also of the awards. It was a fantastic work that they have done over the years. And today, this morning, in fact, I want to say hi to the online people as well. Hi. So I'm going to just briefly introduce you to the field and tell you how the CRISPR-Cas system was actually discovering where we are today. And really, this is the outline of my talk. First of all, I'll go right to the headlines, the technology itself. And then after that, I'll go into the biology of the CRISPR-Cas system, how we can use it to make bacteria more resistant to bacteriophages. And of course, if I have time, because I tend to talk a bit, I'll talk to you about how we are using the CRISPR-Cas9 to edit phage genomes. So, I mean, you are here, you know about the CRISPR-Cas9 technology. If you have been working in biological sciences, you have not lived in a cavern for a while, you should know this is a pretty high topic. So this is, when you type CRISPR in the web of science, this is a number of publication related to CRISPR in the past few years. As you can see, this is going pretty fast. Number of citation as well. When I show the slides to my students, I tell them this is what you call a career opportunity, really. So if you're not using the technology, you should get on board because it's pretty exciting. So the technology really took off, obviously, in 2012 when Emmanuel Charpentier and Jennifer Doudna proposed the use of this technology to edit any kind of genome. And then very rapidly, Fong Zhang and George Church in Boston were able to move this to human cells, and really the field took off. And pretty much everything really has been modified using your CRISPR-Cas9. So you see a list of organisms here that have been modified. Which is really stunning here is the years. Look at the year these organisms have been modified, 2013. So this technology was taken very, very rapidly by the biological scientists. And the reason for that is because it's pretty simple. What you do need is obviously a gene of interest that you would like to modify. Then you will look for a little motif called a PAM motif. I'll show you where and how this was discovered. In Strep pyogenes, the Cas9 will need a NGG motif. Then you will need to design a GPS, or what we call a guide RNA, to really bind to a specific piece of DNA. Then you'll provide some scissors, the nuclease Cas9. And then if you provide also donor DNA, well, the cell, when the DNA is cleaved, will need to repair itself. And if you provide a donor DNA, you can actually introduce a mutation, introduce a gene, delete the gene, and so forth. And then you let the cell do the rest. So this is a little video from MIT that we had edited with their permission, just for educational use, that kind of shows what's going on. So you can go inside a cell. I hope you recognize DNA. If you don't, tell MIT. This is Cas9 that is armed with a guide RNA right here. It will recognize the little PAM motif I was talking to you about. And then by base pairing, that RNA guide will bind to one strand of the DNA. And then, of course, Cas9 will be ready to cleave bluntly the DNA. Then again, the cell as a cut DNA, as a cut genome, needs to repair it, and if you provide a donor DNA, of course, you can repair it the way you want. So that's really how simple this is, and that's why the technology has been used by so many people around the world. What people don't know as much is that part of this was really found in bacteria, as Karen was mentioning, and these discovery came by studying phage and bacteria interactions, and I'm going to go into that a little bit now. But before, I will talk to you about phages a little bit. I don't know if you know about this, but phages are the most abundant and diverse biological entities on the planet. There's nothing more abundant than viruses. It's not bacteria, certainly not humans, animal, plants, it's viruses. Just give you an idea. If you go to the Lake of Ontario, right, take water in the palm of your hand. You have more viruses in your hands than you have human on the planet. All right, so think about this for a second. Next time you go to the beach, all right? So they will outnumber bacteria by 10 times. And essentially, if you have bacteria, you should find phage. We have phage on us, you have phage in you. And really, if you do not find phages, it's because, well, your protocol is probably wrong, all right? So they're really everywhere. And today I'll talk to you mainly, in fact only, about double-stranded DNA viruses. I'm not going to talk about RNA viruses, and I'll also talk only about virulent phages. So I will not talk about phages where their genome can get integrated into the chromosome of bacteria. I will really talk to you about virulent lytic phages. So this is, again, phage 101. So when a phage will recognize a bacteria, so this is a lytic cycle. When the phage recognize a bacteria, this is something what we call, when I teach this concept, I use the term lock and key system. So the phage has the key and the lock is at the cell surface, and you need the perfect match for the phage to actually absorb at the cell surface. And when this does happen, then the phage will actually absorb, inject its DNA, and then start replicating its DNA, producing its own proteins, self-assemble, and after half an hour, for the most efficient phage, the cell will burst and new virions will be released in the environment. And the phage I will talk to you about today are really, really fast. They're like the Usain Bolt of virology, right? When they get inside the cell, they really take over the cell, and that's the end. I guess you could say also André de Grasse for a Canadian sprinter, but that's fine too. So if you're able to actually block one of these steps of the phage replication process, then you will have yourself a phage-resistant bacteria, all right? So phage, very abundant, very diverse. Of course, they play a major role in the ecology, so they control the bacterial population. Some people even think that we may be able to treat or prevent some bacterial infectious diseases using phages. But here, phages are kind of seen as the enemy. Because if you're using a bacteria to produce one of your major protein of interest, and then you have a phage in your culture, then you won't be able to produce that protein because it will kill your culture. And in dairy industry, you actually rely on bacteria to ferment the milk and especially lactose. So if you look at like cheese ingredient, the ingredients for cheese, for example, normally the first ingredient should be milk, well, we hope. And then at some point, you'll see bacterial cultures. And those cultures need to be phage resistant to be able to perform their task, right? Well, unfortunately, those bacteria are often sensitive to phage. And phages are actually obviously natural, and so they can be found in the environment. Then if you get milk, it will be transported into your cheese producing plant. The milk will be pasteurized, you will add bacteria, and if everything goes well, you get some decent product to eat, right? And if there is phage in there and they're infecting it with bacteria, you won't have your product, you will have a low-quality product. And phage, really, we have this joke in my lab where we see a milk truck here. It's kind of a phage truck for us because phage are actually found naturally in milk. They'll survive pasteurization process, so they'll be really present. And if you add the wrong bacteria, then you may have a problem. So luckily, bacteria have multiple ways of defending against phages. They are a system that will actually block the absorption of the phages at the cell surface. There are mechanisms that will prevent the DNA to enter the cell. You get mechanisms that will cut the DNA, like the restriction modification systems, and of course, CRISPR-Cas systems that would fit into that area. So CRISPR, you have heard yesterday. I was not here, but I heard that Rodolphe and Philippe covered this part where the CRISPR really is a genetic structure found in the genome of many bacteria. About half of the bacteria will have a CRISPR-Cas system. About 90% of the archaea will also have a CRISPR-Cas system. So you see the repeats here in the variable regions. And I'm going to talk to you about Streptococcus today. So in Streptococcus, the repeat is very short. It's about 36 base pair. In the variable region, what we call spacers, they're also very short. And in Streptococcus, they're 30 base pair. All right. And it's very interesting because if you look at a CRISPR array of one strain and you compare it with another strain, the repeat will be the same, but the variable region will obviously vary from strain to strain. So it's a very nice strain tracking tool that can be used in microbiology. So of course, there are Cas genes that are next to it. Those Cas genes are very important because this is a CRISPR-Cas system. So Cas stands for CRISPR-associated genes. It will obviously produce proteins, and those proteins will be involved in the three main steps. And the CRISPR-Cas9 technology is actually based on the type 2A system. So it relies on 4 proteins, 4 genes. Those Cas9 protein, the big Cas9 protein is here that is involved in the cutting of DNA, and those 3 genes also involved in the acquisition step. All the structure of these proteins have been solved, and there's a lot of work that has been going on in this area. But in 2005, 3 papers came out, all 3 of them from Europe, saying that those variable regions, those spacers, were matching very often phage sequences or plasmid sequences or foreign element sequences. And they were starting to have some ideas about the fact that this may be involved in some defense mechanism. One very interesting paper was this one from a group in INRA in France, because they were studying Streptococcus thermophilus, which is a bacteria that I've been studying in my lab since 1996 when I came back to Canada. And when I came back to Canada, we have been studying dairy phages for a long, long time. And I'm going to use this as a model to show you how we, with obviously Rodolphe and Philippe, we stumbled into that. So our strain we use is a yogurt strain. It was sensitive to phage, and that strain is sold worldwide to make yogurt. It's very an important strain, give you a very nice texture to your yogurt, so really need to have a bacteria that is resistant to phage here. So the aim was to develop a phage-resistant bacteria, and most importantly, using natural ways. So we had the genome of that sequence, thanks to the DuPont, and then we knew that it was CRISPR-Cas system into the cell. So again, we had been studying phage in my lab. I sequenced my first phage genome in 1998. It cost my lab $40,000 on the NSERC Discovery Grant. This was like crazy or insane, I don't know. Now it costs about $200 to do. But yeah, it was a kind of interesting investment anyway. So we had been studying these phages for a while. We had microarrays data when they were popular at the time and so forth. So we had a lot of information on the phage itself. So this is a very simple slide, but it's the key to the discovery of CRISPR, really. So you got, this is a lawn of bacteria. There's no phage in it. And if you add a little bit of phage, you'll see plaques. These are the results of your lytic cycle I was showing you. If you add more phages, well, the lawn will disappear because the phages will have actually killed the bacteria. If you add really a lot of phages, there won't be any more bacterial lawn. But if you incubate this display for like 48 hours, you'll see colonies that will start to emerge. And those colonies are what we call bacteriophage insensitive mutant. They're generated naturally, and they are resistant to phage. The thing is, in 2005, when we were analyzing those colonies, we could not understand what was the resistance mechanism. It was not fitting any of the known defense mechanism. Turned out that also in 2005, the DuPont has actually filed a patent on the possibility of using this as a defense mechanism against phages. So we could hook up together and try to understand what was going on. So when we took those colonies and tried to understand what was going on, we had these CRISPR papers that came up, a patent from the So we really look at what was going on at the CRISPR array. And this was what we call our eureka moment, because when Hélène Deveau in my lab looked at these colonies, and she amplified by PCR the CRISPR array, she was able to see that the PCR product of the array was actually higher than the wild-type phage-sensitive strain. It was just a little bit. It was actually 60 base pair higher than the sensitive strain. We obviously sequenced that and noticed that it was the acquisition. There was 30 base pair that was actually coming from the phage genome that had been acquired in the CRISPR array, and also the one repeat was actually duplicated. So it was always polarized at the 5' end of the array. And if you're looking at different colonies, the spacers was actually different all the time, or most of the time. So it was really interesting. So again, if you take one colony, you have one spacer that is acquired coming from the phage genome. You've got this repeat is actually being duplicated. And if you take another colony, you've got a different spacer picked up. So when we look at, and you can do this, which is really cool, you can do this again with a different phage. And if you do this, then you'll get again another spacer that will be acquired at 5' end. So you can do this with multiple different phages, and you can get acquisition of spacers. And the more spacer you have, the more phage resistant you will be. So we look at all these colonies and looking at the spacers that have been acquired and wondering why this piece of DNA was actually picked up by the CRISPR array and put in the array. Why was that piece of DNA? Was there some kind of motif that was really important? So we were looking at all these sequences that had been acquired in the array, could not find anything. So we decided to look next to it. So if you look at the phage sequence, what we call the Proto-spacer in the phage genome, so that part that has been acquired into a spacer of the bacteria, if you look next to it, there's this little motif that was always present. That's what we call now Proto-spacer adjacent motif. And that motif is extremely important for the defense mechanism and also for acquiring DNA. And in the genome of the phage that we use, there's 233 PAM motif for that CRISPR-Cas system, meaning that one single phage, you can actually acquire in the same array 233 different pieces of DNA, which is pretty amazing if you think about it. So I know this section is sometimes a bit complicated to understand, but so in the bacteria, you do have the repeat, the spacer, and the repeat, whereas in the phage, you'll have the proto-spacer, same sequence, and next to it, you'll have this PAM region that is really important. It's a kind of also a self-non-self recognition motif. So I'm not going to go through the detail of how this is acquired, but the phage will get in, piece of that space, proto-spacer will be cut and taken, and then will be introduced at the 5' end of the array, and that this repeat will be duplicated. This is going through integrase-mediated event work done by Jennifer Donna's lab. However, if you are a phage biologist like I am, and you're trying to understand what's going on, this kind of didn't make any sense. I was telling you that these phages are extremely virulent, very rapid, right? So how can a phage get inside the cell that CRISPR have the time to take a piece of its DNA, put it in a CRISPR array? For us, it was a timing issue. And we thought maybe something was going on, and we made the hypothesis that probably that the new spacers was actually acquired by the array through a defective phage. So this is what Alex Hines in my lab, a postdoc, did. What Alex did, he did the same experiment, except he added defective phages that had been inactivated through UV or through a restriction modification system. And sure enough, the more we had defective phage, the more resistant colony that appear. Really showing, I don't have the time to go through all the controls, but really it was showing that the more defective phage you're adding, the more phage-resistant colonies you're getting, and the more spacers are actually acquired. So for us, it's really neat because if you think about it, when we get our flu shot, our vaccine against the influenza virus, we get defective phage, or defective viruses that we get for vaccination. It seems that the bacteria has been doing this for a long, long time as well. So it's the same model, except we think this is happening mostly with defective phage. Spacers can be acquired also through a plasmid. So if you put a plasmid into a cell, at some point the CRISPR array can pick up a piece of DNA from the plasmid itself. So the plasmid will no longer be able to replicate in that strain and will be lost by the strain. In that particular paper, it was really interesting because we used this plasmid, low-copy plasmid, and there was an antibiotic-resistant gene on that plasmid. And then we grew the cell without antibiotics for a while, and many cells lost the plasmid. Turned out that when we look at the CRISPR array of these cells that had lost the plasmid, they had acquired a new spacer. And that new spacer was coming from the plasmid, but it was coming actually from the antibiotic-resistant gene. So when we tried to reintroduce the same antibiotic-resistant gene into that cell, we could not. So we had, in fact, created a strain that was refractory to the introduction of an antibiotic-resistant gene. So they tend to like these things over there. So another twist that Alex did recently is that he actually cloned a phage Proto-spacer phage sequence into the plasmid, put the plasmid into a phage-sensitive cells, and then challenge with a phage. So you would expect to get BIMs, and then you would expect those BIMs to have acquired spacer from the phage genome. But turned out that most of the colonies had acquired spacers that we had cloned into the plasmid. So we have this technique where we can introduce any kind of spacer we want, so that's why we call it a spacer on demand. You just need to clone it on the plasmid, put it in a strain, challenge with the phage, and this is the Proto-spacer on the plasmid that will be moved to the CRISPR array. So bacteria can acquire spacers from foreign DNA, such as plasmid, phage genome, and those spacers represent molecular bookkeeping of past infection, and the cell will use that memory to defend against itself, against other phage. This was a fantastic experiment we did in 2006, 2007. Now, since 2014, that adaptation experiment we're using in our undergraduate microbiology labs at Laval Universite, at Universite Laval. This has been a fantastic tool for the students because students love CRISPR-Cas, right? And also it allows them to use the knowledge gained in various areas of their curriculum, like microbiology, virology, bioinformatics, genetics, and so forth. So it's been really a fantastic tool for the students, for our seniors in microbiology. So after the adaptation of the vaccination step, you've got the maturation step, which is the second step. So I'm going to go a little bit faster here, but essentially you've got a promoter here in front of the array. This will be transcribed. It has been shown yesterday. This long RNA will be mature or processed. At the end of the day, you got CRISPR RNAs. Those CRISPR are very short RNAs, between 40 and 60 nucleotides, depending on the type system. In type 2, this was discovered by Emmanuelle Charpentier's lab when she was in Sweden. What Emmanuelle group found is that upstream of the Cas genes, you've got the array, you've got a Cas gene, this little sequence here, which was actually matching perfectly, almost perfectly, the repeat of the CRISPR array. So at the end of the day, you had yourself 2 RNA that was forming an hybrid. Transacting RNA here, here, and you've got matching part of the repeat. So what was going on is that you had in the cell like this surveillance complex, which had a Cas9 protein hooked to the tracer and the CRISPR RNA, which is about 40 some nucleotides. So you got the double-stranded DNA, then your CRISPR RNA, the spacer matching one strand of the DNA, you've got the repeat part, and then the tracer RNA, and it will cut right here. And in the CRISPR editing tool, they actually made a nice little twist where they added a linker here, and you don't have to carry 2 RNA molecules, you just only need 1. So interference step, as Karen was mentioning earlier, we wanted to know how the CRISPR-Cas system was working, so we were interested to understand if it was RNA or DNA to target at the time. So this is a phage-sensitive strain, as you can see. This is a bacteriophage insensitive mutant resistant to 1 phage, and this one is resistant to 2 phage. So we took those cells and look inside the cell. So isolated the total DNA of these cells and look what was happening to the phage DNA inside those cells. And we ran some typical southern blot assay. So this is the strains we use with different spacers that have been acquired at 5' end, and the phage that we use also for these assays, and where the proto-spacers are located in the phage genome. And what was happening is that, so this is the wild-type phage-sensitive cells over time. So you see the increase of this phage fragment, which makes sense because the phage DNA is replicating. But if you look at these BIMs that had acquired new spacers and are infected by a phage again, turned out that it was this extra DNA that was, this extra band that appeared, and we sequenced that band, and we were able to see exactly where the CRISPR-Cas system was cleaving phage DNA. And if you had acquired 2 spacer, it was cleaving the DNA twice. And by doing so, it was Josiane Garneau's work in my lab, we were able to show that it was cleaving 3 nucleotides within the proto-spacer and next to the PAM region. So we knew exactly where it was cutting inside the cell. And this was providing resistance. We knock out this gene, was called at the time Cas5 or CSN1, but became Cas9 because of the new nomenclature. But when we knock out that gene, we could not cleave the phage DNA anymore. So this is another educational video that we adapted from Denisco DuPont. I just summarize what I just told you about. So this is Streptococcus thermophilus in yogurt. Well, you'd like to think it's in yogurt. It's multiplicating and unfortunately will be infected by a phage, will inject its DNA inside the cell, and of course the phage will replicate, produce its own protein, self-assemble, and after again half an hour the cell will burst and new virions will be released. About 100 new virions are typically released in these cells after half an hour. So what can happen though is sometimes phages or defective phages will infect those cells, and then it will be recognized by the CRISPR-Cas system, and it will pick up part of its DNA, introduce it into the CRISPR array, and during that time after that, the cell will start producing this surveillance complex, right? That will have the CRISPR RNA, that will have tracer RNA, that will have Cas9, and this will be ready. If a new phage gets in, it will be recognized by the surveillance complex, and then it will be cleaved by the technology. And then at that time, the phage can no longer repair itself, and that will be the end of the phage, and the cell will actually survive. However, phages are the most abundant biological entities on the planet, right? So they have a few tricks to stay on top. And you probably didn't see that when I showed the first time the slide, but here, this is a little plaque here. These are called phage mutants. And if you look at those and start sequencing them, $200 at that time, it was 35,000 base pair we looked at, only one single mutation into that phage genome. Where do you think it was? It was in the Proto-spacer or in the PAM. So again, the strain we use acquired this piece of DNA, the phage as the Proto-spacer with the PAM, and you've got yourself, you have 3 different mutants that all mutation either in the Proto-spacer or in the PAM motif. So 1 mutation is enough for the phage to bypass the CRISPR-Cas system. Of course, if there's two spacers, then you need much more mutation, and the frequency will be much lower. If you have 3 spacers, then it's very difficult for a phage to bypass the system. And we analyzed a bunch of phage mutant. Turned out that all the mutations were actually localized either in the PAM region or really at the 3' end of the Proto-spacer. So we call this now the seed region. But this fantastic work that has been done here at the University of Toronto by Alan Davidson and Karen Maxwell, well, they actually found a phage that, believe it or not, carries proteins or gene coding for protein that will block the CRISPR-Cas system. So imagine that, protein produced by the phage that will block the system to work. Now, come on, this is fantastic. So the phage will always find ways to bypass the system. And that's why in a bacteria, you'll have multiple defense systems. Otherwise, the bacteria will lose. And that's what we try to do. We try to introduce other defense mechanisms into Streptococcus and make the strain even more resistant. And we did that by showing that the CRISPR-Cas system and the restriction modification system are actually compatible together. And then we are able to really build a robust phage-resistant strain. So, last few minutes. Now we have used the technology to twist it around and try to use it to understand phage a little bit more. Whoa, what's going on? It's my own computer on top of that. Should be a phage genome somewhere in there, right here. Turn out that if you look at the phage genome, about half of the phage genome is made of genes coding for proteins involved in the structure of the phage. And we have pretty much solved the structures of all these proteins. So we have a fantastic idea of what these proteins are doing. But however, this other half of the phage genome, the genes are coding for proteins that have no functions. So for us, it's like the black box, and we don't know what they're doing, and we're really interested to understand what these proteins are doing. So we're using the CRISPR-Cas9 to do that. Case study, we started with a gene and protein called OR47, a very highly conserved protein into the phage we're studying, and we have no function, so we want to know what's going on. So what we did, and Marie-Laurence Lemay did, or started to do, is that she generated a plasmid with the Cas9 gene and a spacer targeting that phage of interest. She provided also on a second plasmid a template or donor DNA with the mutation we wanted to introduce. And that construct is actually providing phage resistance, and when we provide the template, we're able to get mutations. So essentially how it works, when the phage infects, the Proto-spacer will be recognized here by the spacer and the surveillance complex here. It will cut the phage DNA, and because the phage DNA cannot repair, but if you provide it with the donor DNA, it will recombine, and you get yourself a mutated phage. And this may sound simple, but phage biology is very, very, very hard to actually do this, because you need to do this inside the cell. You need to do the mutation when the phage is replicating inside the cell, and we don't have really many tools to do that. But now with the CRISPR-Cas9, we can do this. And we were able to generate a phage mutant deleted from that gene. Now we can study that and see what's going on, what's the function of that particular gene. And there's about 20, 25 genes in that phage genome that we don't know what they're doing, so now we're in the process of mutating all of them, and we're also in processing. We're also trying to add tags. You can add a tag, you can add a his tag to this stuff, and you can start doing pull-down assay again to try to understand the mechanism. So more fun ahead, because again, that's the genome that was missing. We're studying all these phage genes and try again to understand the biology of these phages. So at the end of the day, we were facing an applied problem. We wanted to have better cheese, and because our strains were sensitive to phage, we did basic research, and we discovered CRISPR-Cas9. And then other people run with it and develop this fantastic revolutionary tool that everybody is using now. And I'm here to talk to you about this story, but the fantastic students and postdoc in my lab, and I want to acknowledge them. Josiane Garneau right here, Manuela Villon, Marie-Ève Dupuis, and Alfonso were instrumental to when we determined the cutting site of Cas9. And Deveau at the beginning of the study when she did all these PCR, the CRISPR array, Bruno Martel, we did the phage mutant analysis, Alex with the plasmids assays, and Marie with and the defective phage story, and also Marie with the phage genome editing. And really, I want to thank my fantastic collaborators that are in the audience. Denis Romero, which is here. Denis, we have been working with Denis for over 15 years with Denis Dupont. It's been a fantastic experience with us and for me, and now they're my friends. So sometimes you hear weird story with companies, but industry, university collaboration sometimes, but with us it's been fantastic. So I want to acknowledge Denis, Patrick, and Christophe, which cannot be here today, and of course the winners of Gaintner, Philippe and Rodolphe, which of course have been instrumental with the discovery of the CRISPR-Cas system as the defense mechanism. And on that, I want to thank you, and there should be a picture of my group there, which I don't know what happened. There's a bunch of people. Oh, that's bad. Well, at least the website is there, the picture is there, so you can go. We're studying phage, so my lab is all phage, and we're doing phage research. So if you want to know what we're doing, you can go on our website. I've told you it's pretty much up to date. So on that, I want to thank you for your attention. I'll be happy to answer any question this afternoon, I think. Thank you very much.